Hi, so we are going to do a little bit of MCA review today to help you prepare for the test that you're going to take Tuesday, Wednesday, Thursday. So most of these are things that we've already talked about, but there's a couple of new concepts. I would like you to jot a few notes down kind of as we go through, so please take out notebook, paper, some paper and pencil to jot a few things down. Earlier in the year, we did talk about cells and cell organelles and their functions. And prior to that, or around that same time, we also talked about prokaryotic versus eukaryotic cells. So I want to do just a really quick overview, and you should have this in your notes already from earlier in the year. But some things just to clarify. So some differences between prokaryotic versus eukaryotic. Prokaryotic cells are always bacterial cells, and eukaryotic cells are kind of everything else. So remember, you are made of eukaryotic cells, but so are animals, and so are plants, and so are fungi, really everything except that bacteria and viruses, which we'll talk about in a little bit. Some of the primary differences: prokaryotic literally means pre-nucleus. So our prokaryotic cells, oops, went a little too far. Prokaryotic cells lack a membrane-bound nucleus. Eukaryotic cells have a membrane-bound nucleus. So prokaryotic cells, those bacteria, the genetic material, that DNA, really just hangs around kind of in the center of the cell in a nuclear region, but it's really not held together by that membrane to enclose it. Other differences, prokaryotic cells, as you can see, are always single-celled. So prokaryotic cells are always single-celled. Eukaryotic cells might be single-celled or unicellular. That's just another word for single-cell, unicellular. But they also can be multicellular, because obviously you are multicellular, and so are plants and so on. Prokaryotic cells are much smaller and simpler than our eukaryotic cells. So those are the primary differences that I think you need to remember. You might see that on the MCA test. The other thing, and please don't write all this down, but since we're talking about cells, I just want to mention viruses really quickly. So viruses are kind of their own beast, if you will. They do have genetic material, DNA or sometimes RNA, because RNA, remember, is also genetic material. So they might have DNA, they might have RNA. They're not really considered living, and that's because, as you can see here, they really aren't considered to be cellular, because they're not a complete cell. They're really just genetic material surrounded by this fancy protein coat, and they have special mechanisms that are super cool, that are like that adhere to cells, and then they inject their genetic material directly into cells. They cannot reproduce on their own. They can only reproduce if they inject that DNA inside of other living cells, either an animal or a plant or a fungi or something. So when we get sick with the virus, that virus has found its way into our cells and then begins replicating inside of our cells, and then our cells burst open and spread these virus particles, and then those go in our body and infect other cells. But they're really not considered living, because they really can't reproduce unless they infect a cell that they can reproduce in. And viruses are very tiny. They're smaller even than bacteria. Then a quick review of eukaryotic cells, and I'll probably spend a little bit more time on this on Monday as well. But remember, eukaryotic cells have all these fancy organelles that have specific functions. The nucleus is that powerhouse, or kind of the brain of the cell. Ribosomes make proteins. rough endoplasmic reticulum, transport proteins for export, smooth endoplasmic reticulum. There's some things that are synthesized like steroids and so forth, and it moves those things around. Nuclear pores, or just little like pores in your skin that allow some things have to leave the nucleus, like that RNA, which we'll talk a little bit more about in a little while. Cytoplasm is the gel. Mitochondria, this very specific structure that has those cristae folds on the inside. Mitochondria is for making ATP. Lysosomes are for digesting things. They contain digestive enzymes. Our Golgi bodies or Golgi apparatus, you might see that called either one of those names, is for packaging things up into vesicles, especially proteins that are going to be exported. Centrioles are really only used in mitosis and meiosis. The primary differences between plants and animal cells are, of course, plants are photosynthetic, so they have some very specific structures that are necessary in order for photosynthesis to occur. That's those chloroplasts. So chloroplasts are where, whoops, not sure what I did. So plant cells have those chloroplasts that contain chlorophyll. And that's that magic photosynthetic pigment that allows plants to capture energy from the sun, and along with using some water and some carbon dioxide is actually going to convert that into glucose, which is going to be food for the plant, and it's also going to store some of that so that when we eat the plant, we get that energy as well. Plants also have cell walls. They lack a skeletal system, so having a cell wall is pretty important for structure and support. The other thing they have are these large vacuoles. So animal cells also can have vacuoles, but plants have very large vacuoles that are used for storage of water and some enzymes, but plants have to store a lot of water. They can't just take a drink when they're thirsty like we do, so they have to have that mechanism in order to store that water. Some other things we talked about were how things move into and out of a cell. So just again, a super quick overview, because you may see these on the MCA. How does the cell membrane maintain homeostasis? Remember that the cell membrane controls what goes into and what goes out of a cell, so that maintaining homeostasis begins even at that cellular level. So homeostasis, maintaining internal conditions we talked about. We have to maintain constant temperature, constant oxygen levels, the proper pH, the proper glucose levels, and all of these things. But it all begins really at that cellular level. It has to control what goes into and out of the cell. It especially begins with carbon dioxide and oxygen diffusing into and out of cells. You remember, when you breathe in, you don't breathe in because your lungs need to be filled up. You breathe in because you're getting oxygen into your lungs so that that can diffuse into your cells, and that carbon dioxide can diffuse out of your cells because there's a concentration gradient as your cells use up oxygen. They have an excess of carbon dioxide, so it has to get rid of that. It has to bring oxygen in and get rid of carbon dioxide. Diffusion is very simply occurs because of a change in concentration on either side of a membrane. So things will diffuse if there's a concentration gradient. Earlier in the year, I demonstrated that I dropped a little food coloring into a beaker of water, and we watched as it dissipated through the beaker. I said, just like when some kid pees in the swimming pool, right? There's pee everywhere because that's going to dissipate throughout the pool. That's a form of passive transport, because passive transport doesn't require energy. If it's moving along with that concentration gradient, that really doesn't take any energy. It would take a lot more energy if you wanted to get that food that food coloring that dissipates throughout the beaker, if we want to somehow get that out of there to coalesce it back into a single drop of food coloring, would require some energy. That doesn't happen passively. That would require some sort of active or energy use. So in cells, active transport uses energy. It's either moving particles that are very large through the cell membrane that can't diffuse through or can't easily move through the protein channels, as is the case with endocytosis and exocytosis. So endocytosis are moving large particles into the cell. Exocytosis is taking large particles out of the cell. The other type of active transport is if it's moving it against a concentration gradient. So like I said, if we had that food coloring and it was all dissipated in the water, and then I wanted to somehow move it back out of the water, that would require the use of some energy. That would actually be active transport if it went against the concentration gradient. We demonstrated kind of osmosis, which is movement of water through a membrane, when we had our eggs, and we put eggs in vinegar, and water kind of moved into the egg because the solute level was higher inside of the egg. We put an egg in corn syrup. The egg actually got smaller because the solute level was higher in the corn syrup than it was inside of the egg. If we found the perfect solution, which we would say is isotonic, it would mean the level of concentration, or the concentration of solutes would be equal on both sides of the membrane. So that would be an isotonic solution. And again, if that water moves through a membrane, if a solution gives up water, we call it hypotonic or water fearing. Hypertonic means water loving. It would move, it would attract water to itself. And then isotonic is when it is equal concentrations on both sides of a membrane. I also want to just really quick reminder, DNA and the way that DNA, remember that DNA never leaves the nucleus, so it has to like code those, it has to carry the code for the proteins that are made in our body, but it has to transfer that code onto RNA. So the rule always, and I'm pretty sure you're going to see this on the MCA, this is super important. DNA copies its code onto RNA, which then translates that code into actual proteins. So the DNA just stays in the nucleus, but it just makes copies of itself. And then it makes copies of itself to make more DNA if the cells are going to divide. But it copies its genetic code onto the RNA, and it's really that RNA that goes out, tells the amino acids what in what sequence to assemble so that it builds the correct protein for your body. And your proteins are different than my proteins, a lot of them, because that's why we look different, right? Eye color, hair color, skin, but even the proteins that make your tissues and all of that. Always remember that that general structure. So the guanine pairs with cytosine, cars in the garage. Adenine pairs with thymine, apples grow in trees. Everybody likes sugar. These pair onto the sugar molecule on that, what we call the backbone, that sugar phosphate backbone of the DNA. And you guys built a model of that in class, so you should remember that. And here's my less than a minute overview of mitosis versus meiosis. So mitosis, always remember mitosis happens in my toe or in the body cells. It makes body cells. It's one round of cell division, resulting in two absolutely identical daughter cells, because one has made an exact copy of itself, and now there's two. It's replicated the DNA. It's gone through the stages of mitosis. And then cytokinesis, the final cutting of the cell. Mitosis is really necessary for